Good afternoon, everyone. So today we are going to discuss about the endoplasmic reticulum, which is the most important structure that we found in the cell, any living cell, and it has a very important function in the protein translocation. So today we're just going to have a brief overview of how ER looks like, and then we will just be watching a short video. In that we will be getting a clear idea of how protein synthesis works. So let's start. So basically, endoplasmic reticulum is a membranous structure which we have in all eukaryotic cells. It's a network labyrinth of branching tubules and flattened sacs. It has the phospholipid membranes which separates the endoplasmic reticulum lumen from the cytosol. The most important functions of ER varies greatly upon cell types, but the most important function that ER has is the protein synthesis and lipid synthesis. So there are two different types of ERs. One is rough ER. The rough appearance is because of the ribosomes. Now you can easily understand that it's rough is ribosomes, and ribosomes means it's going to synthesize the proteins. So this is typical structure of endoplasmic reticulum. We have a nucleus over here, and you can see this is all the endoplasmic reticulum that we have. If you can see just the nucleus, once we leave the nucleus, immediately we have a rough endoplasmic reticulum, and then we have the smooth endoplasmic reticulum. So the reason behind this is, mRNA which comes out from the nucleus will easily comes into the endoplasmic reticulum, near the endoplasmic reticulum, and it will start the process of translation, which in turn produces the proteins, or we can say the chain of amino acids. So this is what we're going to discuss now. So protein biosynthesis is the most important process that we have. So we'll just play a short video and we can discuss in that actually how the protein synthesis looks. So this is your 5' to 3' RNA, mRNA, which will start the process of translation through the process called as a polyribosomes. And these polyribosomes are very important for the protein synthesis. So these are actually in the subunits, and what will happen, they will just take the entire machinery towards the endoplasmic reticulum membrane. So this is our endoplasmic reticulum membrane. This is the cytosolic part, and this is the ER lumen part. So we'll now go further. So as you can see, this is cytosol, this is ER. Whatever water soluble proteins are being produced, immediately when they are produced, they are taken inside the endoplasmic reticulum. Some transmembrane proteins are also synthesized in this particular way. So what we have just now here, if you can see this, we have a signal sequence, and this signal sequence is basically a sequence which is present on the emerging proteins, which is very important, which is identified by the signal recognition particle, which are already there in the cytosol. So once they identify the signal sequence on the emerging polypeptide chain, they will go and bind to that, and this particular SRP will take and go and bind to the SRP receptor which are present on the endoplasmic reticulum lumen. And there are some pores or translocators which will start the polypeptide chains to get inside the endoplasmic reticulum. So just keep in mind that this signal sequence, the role of this signal sequence is to take the entire machinery or polypeptide chain towards the surface of the endoplasmic reticulum. Once it is done, the purpose of the signal sequence is over. So in that case, the signal sequence will no longer be required. It will stick to the translocator, and entire protein will be taken inside. And once the proteins are synthesized, once the entire protein or polypeptide chain comes inside, the enzyme called as a signal peptidase will cleave the protein from signal sequence, and in this case the translocator will be closed. So this is how a specific or typical protein biosynthesis works, and the plug will be taken back to the translocator, thereby not allowing anything inside which is non-specific. And that is the most important thing, because if you have something which is coming inside the endoplasmic reticulum, that is not good for the cell. So this is how a typical protein biosynthesis works. So I hope in this particular short lecture you have understood how the protein biosynthesis works, what is the structure of endoplasmic reticulum, and in the next lecture we'll be discussing about different functions of the endoplasmic reticulum. So till then, take care, thank you very much, and keep watching.